Hello, everybody. My name's Melody, and I'm here to talk about genetics, more specifically, genetic engineering. Scientists have been expanding very rapidly on the field of genetic modification for the past few years. After scientists discovered DNA and how it works, they started to do more experiments. Each discovery improves our understanding in medicine, agriculture, and research. And there are lots and lots of accomplishments, such as radiation to cause mutation, GM crops, faster growing chickens. And my topic for today is specifically going to be about genetic engineering in the world of dogs. As a dog lover, I try to know more about dogs, not just different breeds or how to take care of them, but what experiments scientists do to change and transform the species Canis lupus. The experiment I'm going to talk about today is how Chinese scientists use gene editing technique to produce the world's first genetically engineered dogs. Let's first talk about a species of dog that originated from England, the Whippet. Whippet is a medium-sized dog that can run really fast due to their thin long legs and also a relatively thin medium-sized body. They become really popular for dog racing because of its agility. They have very few health problems, but there's one kind of genetic disease that only Whippets can have, the bully Whippet syndrome. What's a bully Whippet? The one on the right is a normal Whippet, and this is a bully Whippet. Stronger, bulkier with muscles, and it even has 8 packs on its stomach. And you can really see one on the road. It has double the amount of muscles, and you might wonder, what is wrong with them? The bully Whippet syndrome is a genetic disease manifested by a mutation in the myostatin gene. Myostatin is a kind of restriction enzyme that restricts muscle growth, and without the myostatin gene, the Whippet will have increased racing speed and double the amount of muscles. This mutation can only happen in Whippets. So how did it happen? Now I'm going to use models to explain. Connecting to what we have learned before, there must be an error in the dog's DNA in order for a Whippet to have a genetic disease. This is the dog's gene, and this part codes for the myostatin enzyme, which is a restriction enzyme that restricts muscle growth. But because of mutation, the base pairs are then changed. This mutation inactivates the myostatin. When both copies of the genes are mutated, it is called homozygous deletion. RNA polymerase will transcribe the mutated DNA strand to produce mRNA. mRNA will then go through the sites of ribosome subunits with rRNA to be translated by tRNA. After the codons are transcribed on the mRNA, polypeptide chains are produced. These polypeptide chains are in a specific sequence that codes for different proteins. Normally, myostatin are being produced, but because of the single mutated DNA sequence on both genes, cause the Whippet to lack myostatin, which muscle growth is uncontrolled, and this causes a bully Whippet to be produced. And scientists in China say they can now engineer the same change into other dogs. Similarly, scientists use a gene editing method called CRISPR-Cas9 to knock out the myostatin gene in beagles, making them grow extra muscles. Their objective was to damage both copies of the myostatin gene, which is also cause homozygous deletion, so that the beagle's body would not produce any of the muscle-inhibiting protein that the gene manufactures. So what's CRISPR-Cas9? CRISPR-Cas9 is really precise, cheap, and efficient. It is now widely used in scientific research. Derived from bacteria, the system uses a guide RNA to direct an enzyme Cas9 toward a target gene and cut to disrupt them. So now here's this video about what's CRISPR-Cas9.
When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA. one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target.
So the Chinese scientists engineered CRISPR-Cas9 system to target the myostatin gene on the beagle's DNA. The green strand is the guide RNA. Scientists can change the guide RNA to match and target the bases that codes for the myostatin gene. And the CRISPR system can cut or knock out the gene, because Cas9 is like a scissor that cuts and disrupts the gene. So myostatin won't be produced, similar to the Bully-Whippet syndrome, without the myostatin gene to regulate muscle growth. The dogs will eventually grow more muscular. The scientists injected 35 beagle embryos with their designer CRISPR. Out of 27 puppies, only 2 puppies were found to have the desired knockout mutation. So this is Hercules and Tango. So these are the 2 puppies that have the knockout mutation. The dogs didn't quite become the ultra buff like Hulk beagles you may have pictured. Hercules still managed to produce a small amount of myostatin, but significant enough to make him different from other dogs. Tango, however, was more muscular, showing beefed up thighs. And why did scientists do it? Why do we need muscle dogs? There must be some advantages. The first reason is for police and military force. The second reason is to study similar disease in humans. So with great muscle mass, the dogs can hunt, and they are expected to have a strong running ability, which is good for hunting police applications. Later on down the line, scientists would like to manipulate the dog genome in order to mimic and copy human diseases. To your surprise, this change can happen to humans. About 10 years ago, a German child was born extramuscular as a result of being born without the myostatin gene. Dogs exhibit close similarities to humans in terms of metabolic, physiological, and anatomical characteristics, which makes them an ideal genetic models to study human diseases. With all these advantages, there are definitely some disadvantages. First of all, it's unnecessary. Second, the balance in the dog's body can be harmed. Third, the ethics of making designer dogs. So according to this graph, the violent crime rate in the United States from 1990 to 2015 is decreasing. Also, the total violent victimization went down 72%, and serious violent victimization went down 75%. So is this really necessary to have mutated dogs as military troops? Second of all is homeostasis. So homeostasis is the tendency to maintain a stable, relatively constant internal environment in the dog's body. So the dog's body won't have the ability to maintain homeostasis because they need to use too much energy for their extra muscle mass. And also more issues. With those extra muscles, other systems in their body can't sustain it, such as cardiovascular, the heart and the bloodstream, and skeletal systems. Also, reproduction issues may happen due to their unusually heavy and bulky offspring. Also, error can still happen because CRISPR-Cas9 is imperfect. So if one dog has the disease, it may affect generations after generations of the whole canine species. And third, ethics. It's important to think that is it ethical to make designer dogs? Here's a quote from MIT Technology Review. Man's best friend is now his newest genetic engineering project. So dogs are animals, and they are not our tools. If there are no restrictions, people can start to create more and more designer dogs. And using the same technique to create dogs with favorable traits for other purposes. For example, even better smells for airport work, enhance their senses to help handicapped people, and also to help trapped person. And this can cause a huge problem. Mistakes can always happen, causing other mutations such as interbreeding. For example, if we want bulldogs to have shorter legs and noses, it causes them to have spinal problems. So to me, the disadvantages really outweigh the advantages. First of all, it isn't necessary. And second, the dog's body can't function properly with the change. And also considering about the ethics, I just can't quite accept the fact that scientists are actually giving a kind of genetic disease to more dogs. If they keep continuing to do this without thinking about the possible issues, this will really cause a negative change in the canine species. These are the sources that really helped me on this project. And thank you for watching. I'll see you next time. Bye!